Testing 1, 2, 3. Hello everyone, and welcome. My name is Yi-Chung Chung. I'm the academic program manager here at the Hatfield Marine Science Center, and I want to welcome you back for the Ignite Talk portion of the afternoon. So prepare to be illuminated. I hope you enjoyed the poster session, and all of you have voting ballots for the best poster in Ignite Talk. Again, there is an award for best poster and also best Ignite Talk, which you're about to hear. So please fill out the ballot and turn it in. Today's oral presentation format, these Ignite Talks I was just sharing with Cinnamon, actually came from a discussion back in the day with Janet Webster, who was our librarian at the Gwynn Library. And we were talking about how we could develop another format for communicating science to informal audiences. And this was one of those ways, these Ignite Talks. And again, so these Ignite Talks or lightning talks will be short, fast-paced talks from students about their research and why it's important. Okay, and this year we have 12 graduate students who are presenting from several different groups: OSU's Comparative Health Sciences, OSU's Fisheries and Wildlife and Conservation Sciences and Education, Integrative Biology, Marine Resource Management, and Microbiology. I'd like to invite our first speaker, which is a recording, Caitlin Stoner, who is a PhD student in Fisheries, Wildlife, and Conservation Sciences, mentored and advised by Don Lyons, who received the 2022 Holt Award.
Hi everyone, thank you for coming today. My name is Katie Stoner and I'm a PhD student here at Oregon State University. And I want to know what you would answer if I asked you this question: where are the puffins? Would you say out on the water, north at Haystack Rock in Cannon Beach, perhaps on the back of the Lincoln County bus, or next door at the Oregon Coast Aquarium in their aviary? You may have even countered with a question and asked me, why should we care where the puffins are? And to that I would answer that seabirds in general can indicate marine health, connect different regions through their migratory behaviors, and they are important components of marine food webs. But puffins specifically are some of the most well-known seabirds, with their colorful plumage and interesting behaviors. And the answer to where the puffins are depends on where you are in the world and at what time of year you ask this question. There are 4 species of puffin, 3 of which are found in the Pacific and 2 of which are found in Oregon, rhinoceros auklets and tufted puffins. And tufted puffins are probably the most well-known puffin in Oregon. Puffins are iconic seabirds that are linked to the identity of the Oregon coast, as you can see statues along the coast, and they're linked to marine environments in the North Pacific. Perhaps the most well-known and largest tufted puffin colony in Oregon is located on Haystack Rock at Cannon Beach, but it is getting more difficult to see puffins on the Oregon coast every year as populations of this species decline. However, population sizes of tufted puffins are changing differently in different parts of their range. They are increasing in size in the Aleutian Islands, likely decreasing in the Gulf of Alaska, and dramatically declining in the California Current. There are projections that the California Current, the region that includes Oregon puffins, could lose its tufted puffins as soon as 2050 due to loss of suitable nesting habitat for the species in this region. Puffins' secretive breeding habits and underground nests that are located on offshore islands make population changes difficult to assess, and we often can't confirm declines until populations are very small. But studying puffins in areas where they are likely declining but still present in large enough numbers, such as Kodiak Island in the Gulf of Alaska, allows scientists such as myself to examine possible causes of declines. We have a relatively comprehensive understanding of the breeding resource and habitat needs of tufted puffins during the breeding season. For example, how many eggs they lay and where they dig their burrows. And this also includes knowledge on the prey types and diets that they require to feed their chicks as you can see an adult tufted puffin in this video coming in to provision its chick with forage fish. Because we know the general breeding ecology of the species, we can identify possible threats during the breeding season, such as climate change, oil spills, fisheries bycatch, predation, invasive species, or human disturbance. However, we have not identified one smoking gun cause of declines for tufted puffins during the breeding season, for which to target mitigation or management actions to help stop declines from occurring. And our understanding of threats is complicated by our lack of knowledge of the winter ecology of tufted puffins when they spend 9 months at sea somewhere out in the North Pacific. We don't know where or what they're eating during this time. And this lack of knowledge limits our ability to assess threats and predict how puffins might respond, for example, to competition with other large fishes such as Pacific salmon that winter in the same locations and eat similar food. We also don't know how individual birds might cope with changing environmental conditions, such as harsh winter storms or marine heat waves that may increase in frequency, and both of which can cause adult mortality. Puffins are long-lived seabirds. They can live more than 30 years. And so these threats that impact adult survival, especially during the winter, will have more drastic outcomes for populations than impacts on breeding success in a single year. And so I ask that you reflect on what you feel when you see a tufted puffin. Ask questions about these iconic species because we may know less than you think. What would the Oregon coast be like without these fascinating clowns of the sea?
All right, so sorry. So our next speaker is Charles Nye, who's a master's student in Fisheries, Wildlife, and Conservation Sciences, mentored and advised by Scott Baker, who received the 2021 Markham Symposium, or Markham Award.
Okay. Hey everyone. Oh. Test. Yes. Hi, everyone. Thanks, Yi-Chung, for that wonderful introduction. My name is Charles Nye. This is my Ignite talk, A Gut Feeling, Identifying Gray Whale Prey Using DNA Metabarcoding. So I advertise myself as a conservation geneticist. So basically, I like to use genetics to address questions about conservation. And one of my study species are gray whales, specifically a small subset of the Eastern Pacific population of gray whales, known as the PCFG, the Pacific Coast Feeding Group. If you've gone whale watching off here in Depot Bay, off Vicuña Bay, you've likely seen one of these guys during the summer months. And Hatfield, the Marine Biology Institute, and our collaborators would be remiss not to research 300 whales in our backyard that come every summer. I wonder what they're doing here. And this is where the F in feeding comes in, because gray whales are what we call benthic foragers. They scrape at the bottom of nearshore waters for food, and principally they're believed to feed on mysid shrimp. These are little shrimpy things that live on the benthos. But how do we know this? Well, visual assessments. If you find a whale and you see a whale going after a patch of prey, great. But what if you can't? What if that's hard to do? What if we could use DNA to confirm these observational data? And that's kind of where I come in. And with this fancy tool called metabarcoding, but we'll go first into why genetics is important in marine biological research. It's hard to study the ocean. Hot take, I know. But we don't have gills. Hard to observe things that don't want to be seen. Hard to observe things that are eating other things that don't want to be seen. And so these genetic methods can help elucidate some basic questions like, what are things eating? So think of DNA barcoding as if I took a fingerprint of myself, and that is me, that's Charles Nye. DNA metabarcoding is if I took every single one of your fingerprints and analyzed them at the same time, but genetically. And one very important implementation of metabarcoding is an environmental DNA, but we also have something called dietary DNA. We can get the prey or the stomach compositions or the intake on a genetic level, a species level. I don't want to beat around the bush too much, but we get that from poop. So one great place to get dietary DNA is poop. And our collaborators with the Geospatial Ecology and Marine Megafauna Lab have been collecting gray whale poop off the Oregon coast for a while now. It's been since what, 2016. This was originally a hormone and isotope study, but now we could do genetics on these poop samples to see exactly what these gray whales are consuming. And so some very brave people collect the poop. I am not good on a boat. I get seasick. And so after they get it, I receive it and I filter the poop. I extract the DNA. I do the magic on it to make the magic happen. And what does that look like after we extract and process all that information? Coming up, and after that 15-second marker goes off, a kaleidoscope, I like to call it. So remember those mysid shrimp? They're in pink. So yes, gray whales are eating mysid shrimp. Thank you very much. No, they're eating a lot more than that too. If you look, like I said, it's a kaleidoscope. And coming up shortly, you'll see other prey species that have been identified through gray whale dissections, as well as direct observations at the ocean floor, including olive snails, including those shrimps, including those krill, and including something called ghost shrimp that live in estuaries. But let's talk about the barnacle in the room, and that is the collateral damage. Gray whales are eating a lot more than just their target prey because they're kind of like ocean vacuums. They target patches and they consume whatever is in that patch, prey and other things included. What does this mean for the whale? We're still working on that. But if you look at this, we have the IDs of the prey. Now let's do some science on it. And over time, we can look at the compositions and see that they don't change all that much. And years are a good way to collapse on bigger oceanographic events. We can pay attention to prey proportional signals. And just as a demonstration, look at 2020, that mysid shrimp signal is demonstrably much lower. Why? We're still working on it. Come on. And we can see this change over time as well with a much more complicated way of assessing these community data. But if you look here, that big red circle in the bottom right corner, that is the centroid for the 2020 data, basically the kind of average to where all those data points coalesce together. And we see that it is indeed demonstrably different. Why? Again, we're still working on it, but these trends can be tied to other oceanographic events, and we might be able to patch in as to why prey availability might change, or why whale preferences may change as well. Major takeaways: gray whales are eating a whole lot of different things. Of all the prey items they're going after, it's mysids who reign supreme. Of all the collateral, it's barnacles. Annual differences do exist, and we still have a lot more work to do, and I'm very excited to share that with you, hopefully in the future. Thank you very much to the Markham, to all my funders, my committee, my homies, and my lab itself, and most especially you. Thank you.
Wow, great presentation. Our next speaker is Grace Roa, who is a master's student in Fisheries, Wildlife, and Conservation Sciences, advised and mentored by John Chapman, who received the 2021 Markham Award as well.
Okay, my testing? Yeah. Thank you, Yi-Chung. A wonderful presentation by Charles. Mine is going to be, let's see. So I am talking to you about the final countdown of the age and growth relationship of Eupogibia pugicensis and its non-native parasite Orthione graphenis. So to get started, Eupogibia pugicensis is a burrowing mud shrimp that previously occurred in nearly all of the East Coast estuaries from Morro Bay, California to Prince William Sound, Alaska. These shrimp make these really beautiful permanent Y-shaped burrows in the intertidal mudflats and are incredibly important ecosystem engineers that provide habitat and oxygen to the different critters living in the estuaries in typically hypoxic environments. However, the mud shrimp populations have been declining range-wide, and for the last decade or so have been associated with this introduced Bopirid isopod Orthione graphenis. So Orthione were introduced from Asia in the 1980s and were most likely from ballast water. And they're able to take enough hemolymph or blood from the reproductive shrimp to effectively castrate the female shrimp. We also know that after the isopods settle, so this first peak here, they live to be about 3 or 4 years. And they also infest shrimp above 15 millimeters, which is reproductive size shrimp. However, direct measurements needed to really resolve how or if this parasite or some other ocean or sediment factors are causing these declines have remained elusive because the shrimp they don't really like to be handled, okay? So we can't really deal with them, and so it's hard to see if these isopods are really the reason for the decline. But luckily, the biggest population is here on Idaho Flat, right outside of Hatfield, where we can monitor them over time. We can build a model to see the population dynamics of this relationship through, through repeated observations. So in order to make our model, first we look at the size structure of the average size structure of the female mud shrimp from the last 15 years. And we need to fit curves around the 5 population dynamics parameters. So that's, and, I'm nervous. But remember, the shrimp they only brood once a year and recruit once a year. And so we call that a cohort. So the model has to explain how we get this funky shape where there are lots of little tiny shrimp, only a little bit of the medium-sized shrimp, and a whole bunch of the larger shrimp. This low middle abundance that you see can only happen by females growing quickly through those little sizes and then slowing down as they age. This is also known as the von Burton-Laffey growth pattern. So we know that this first cohort here on the left are brand new shrimp. They just settled in year 0 and they don't become reproductive until they're 2 years old. We also know that the Orthione live at least 3 years old. So that means the shrimp live at least 5 years old, right? So now our model can look for at least 5 cohorts in our population. The problem though is making these cohorts increase in age by 1 year and decrease in age or decrease in abundance each year, but still grow with that same von Burton-Laffey growth pattern. So in order to get our model to work, we add up all of the cohorts and they have to add up into the shape to kind of follow the pattern of that blue line. And that's what we want to see. That gives us our model. But we can test our model's accuracy on how well it's predicted from the size distributions and how they add up, and we can compare them from the observed population to the predicted population. See how that goes. So far, it's pretty good. But ultimately, Eubogibia are recruitment limited, and Orthione take the biggest and the most reproductive shrimp of all. And now the really question is if the population can keep going. It depends on the female shrimp. But honestly, the way that things are going, and if it really is the Orthione, the shrimp are declining at about 2.5% each year, and we only have about 10 years left with them. So please care about my shrimp. I like them a lot, and I want to thank the Markham family for helping me and my committee, and especially Yi Chung for everything that he's done for me, because he's done a lot. So.
You did it.
It's not true. She did all the work.
Great job, great presentations. I just want to state our students are really amazing. Jeffrey Walker is our next speaker, master's student in Fisheries, Wildlife, and Conservation Sciences, mentored and advised by Kathleen O'Malley, received the 2021 Markham Award and the 2022 Jones Award. Great job.
Hello, everyone. I'll be talking about my now completed thesis research using genetics to investigate genetically distinct groups of Pacific albacore tuna, or at least as much as I can fit into a 5-minute presentation. As I mentioned, this research was funded by the Mamie, Markham, and Walter G. Jones Award. So albacore tuna are a highly migratory open ocean fish capable of strong bursts of speed as well as ocean-wide migrations. Of course, albacore are also a great source of lean protein and are harvested around commercial fisheries throughout the world. Albacore harvest is greatest here in the Pacific Ocean, where Japan, the USA, China, Taiwan, and many others manage extensive fisheries. Oregon is also home to lots of albacore fishing, with Newport generally leading the way in Oregon albacore landings each year. Currently, Pacific albacore are managed as 2 stocks in the Pacific Ocean in the north and south, separated by warm, oxygen-poor equatorial waters. This stock delineation has been supported by limited tagging data and low catch per unit effort near the equator. However, there are still many uncertainties regarding albacore distribution, with relatively little known about spawning locations, migration routes within hemispheres, whether migration is occurring across the equator, and whether there's population subdivision between these delineated stocks. And recent genetics projects have searched the albacore genome and identified genetic markers that are different in fish from different regions of the Pacific Ocean. And because of their differences, these markers can be used to determine where a fish likely originated. Our research uses a small subset of these informative markers, which allows us to easily sequence many more fish. So we sequenced a large number of Pacific albacore at these informative markers to address a few main questions. And we first investigated genetic differences between albacore caught in the USA in 2020 and 2021. We then identified genetically distinct groups of albacore using 4 sample areas in the Pacific Ocean and identified migrants among those genetically distinct groups. And lastly, we evaluated the ability of a genetic sex marker to accurately identify sex using just DNA, which could provide a cheaper, faster, and non-lethal alternative to current sex identification, which requires dissection of the fish and observation of the gonads. So from 2020 to early 2022, we coordinated sampling effort throughout the Pacific Ocean by reaching out to collaborators to help collect tissue samples. And I was also able to collect some fin clips myself right here in Newport as the fish were landed. Over 1,800 samples were collected in total from the areas you see here. Most of these were from the USA since they were the most accessible, but we aim to have sampling coverage on both sides of the North and South Pacific so that we'd be able to look at inter-hemisphere differences. We extracted DNA from each sample and then amplified the target markers, which creates thousands of copies of them and makes them easier to sequence. And finally, we sent them out for sequencing, which is where we actually read their genetic code and see how the individuals are genetically different. We tested albacore caught during the 2020 and 2021 fishing seasons off the coast of the USA, but found no significant genetic differences between these years. So as a result, these years were then combined for all of the further analyses. Our subset of genetic markers was able to capture genetic differences between our 4 sample areas. The 2 North Pacific sample areas, the USA and Japan, were the most similar, but subtle genetic differentiation still existed between those 2. Genetic differentiation was much clearer between the North Pacific fish, those from New Caledonia, and those from French Polynesia. We identified these 3 genetically distinct groups in the Pacific Ocean, and then used each fish's DNA to see which area it likely belonged to. Using this approach, we identified 2 migrant fish out of the 1,525 fish we tested, suggesting that dispersal between these sample areas may be limited. Both likely migrants were caught near New Caledonia, but they appeared to originate from different areas. And lastly, we identified genetic sex using a previously established assay as well as our sequencing approach. And our approach was successful in matching the established assay. However, both yielded accuracies around 77 to 79%, which was lower than anticipated. So one key takeaway from this is that the subset of informative markers we used successfully captured regional genetic differences in Pacific albacore and identified sex as accurately as the previously established assay. So this subset can be used in future studies as a standardized method. And a second takeaway is that the 2 South Pacific sample areas showed significant genetic differentiation and limited dispersal. Future research may investigate whether these 2 areas represent 2 separate populations and should be managed as such. So I'd like to briefly thank all our collaborators listed here, and of course the Mamie Markham Research Award and Walter G. Jones Fisheries Development Award, which were instrumental in funding this research. Thank you.
Great job. All right. Wow. Our next speaker is Jess Schulte, PhD student in Fisheries and Wildlife and Conservation Sciences, mentored and advised by Taylor Chappell. Jess received the 2021 and the 2023 Markham Award, as well as the 2022 McNeil Award.
All right. I just want to point out my glow-in-the-dark shark shoes since while we're here. All right. Hi, everyone. My name is Jess Schulte, and I am a second-year PhD student in the Chappell Big Fish Lab, and I'm going to be talking about incorporating apex predators into our understanding of the Pacific Northwest ecosystems. And why are we doing that? It's because predators play important roles in ecosystems. And there's tons of research around the world showing the important roles that they play, as well as the impacts of when they're removed. And that applies right here in the Pacific Northwest. So there's a ton of research that's been done looking at predators here and the coasts of Oregon and Washington, and looking at the way that they fit in the ecosystem and the species that they affect, including salmon. However, there's a missing piece here. Anybody? Sharks, you got it. So that's a lot of what my research is looking at, as well as my lab's research. We're looking at how sharks fit into this ecosystem, and what are the prey species that they're targeting, where are they going, kind of along that information. So Willapa Bay is a large bay located in southern Washington. It is home to a bunch of important prey species, including salmon, as well as Dungeness crab. There's harbor seal there, there's sturgeon, lots of oysters, and sharks. So this is 2 separate white sharks that were caught in the 40s in Willapa Bay. The one on the right, they cut open and they found a ton of Dungeness crab, apparently just Pac-Manning them across the bottom of the water. This is a thresher shark that was found near to Willapa Bay. Sometimes they're seen inside the bay. But both of these shark species are species that are very transitory and not super abundant. And so one of the abundant species that we have that we see along the coast and in Willapa Bay are the broad-nosed seven-gill. So these are a large and abundant apex predator in temperate ecosystems around the world. And I say apex because they're eating all levels of the ecosystem. When they're small, they're eating small fish and crabs. And when they're large, they're eating marine mammals, as well as other sharks and rays. And they exhibit really high seasonal site fidelity. So that means that they are out on the coast the rest of the year, and then they come back to the same location seasonally. And so here on the Pacific coast, that location that they show up in is Willapa Bay. So that leads to my project. I am incorporating their movement, aka where they're going, with their foraging ecology, aka what they're eating, to quantify the impact that they're having in these ecosystems. So what I've been doing is going out into the field into those seasons of high abundance, and I am catching sharks and I'm sampling from them size and sex data, as well as different biological samples that I'll get into. That's us releasing a shark in Willapa. And so to track their movement, one of the things I'm doing is tagging them with acoustic tags. So it's about the size of your thumb, and it emits a unique ping that I can identify the animal as they pass by acoustic receivers. That's obviously not here. If anyone's been on the water here, it never looks like that. And so luckily in Willapa Bay, there's an established array that's there of acoustic receivers. It's maintained by NOAA. And so we can see when our sharks are coming into the bay, as they move throughout the bay, and then as they leave at the end of the summer. Another thing that we're doing, like I mentioned, is foraging ecology. So we are collecting blood and muscle samples, and we're using stable isotope analysis to give a picture into the broader aspect, a long-term foraging ecology. So that gives us weeks to months kind of what they're eating over that time. And then over the short term, we are collecting stomach content. So we are lifting them up onto the boat, tilting them sideways, and then I'm collecting their throw up in a bucket because science is weird. And so if you're like my advisor, you're probably wondering, what have I done so far? And the answer is we have sampled from 92 individual animals. 3 of them have been recaptures. 1 of them was a recapture by a fisherman all the way down in San Diego. So they are potentially swimming really, really long distances. This is a size distribution of all the animals that we've caught, and the brackets indicate the estimated sexual maturity. So you can see that females on the left, typically we're catching immature females, and then we're typically catching mature males on the right. I also talked about collecting stomach contents. And so one of the more exciting stories that happened is we caught a small female, and we would expect her to just be kind of eating small crab and fish on the bottom. And she puked up a perfect chunk of harbor seal, indicating that these animals are eating much higher on the ecosystem than what we originally expected. Another exciting aspect of the project is funded by OSU's Marine Studies Initiative, and it's to bring Indigenous students out on the boat to increase diversity in shark science. And so the middle picture is an art project that was one of the students. She did a beading project of seven gills. And then the right is a video of POV that one of the students participated in over the weekend. And so for the next steps of my project, I'm currently in the second season of my field season. And so I'm continuing to collect those samples and stomach contents, and then I am going to be processing those samples after this summer and incorporating that quantifiable data into ecosystem models to understand how these sharks fit into the ecosystem. And that's it. Thank you to all my funders. Thanks for being here.
Great presentation. And our next speaker is AJ Malazzi, who is a PhD student in the Fisheries Wildlife and Conservation Sciences Department, mentored and advised by Kelly Bidenwig and Sam Chisholm, who also has a guest, Omar, who is from the Siletz, who is a student from the Siletz High School.
Yeah. Ready? Where's ready is we're ever going to be. Hi. H2O Research is a pilot program that's funded by the McNeil Award. Thank you very much. We're hoping 15 seconds is enough for you to appreciate our original title and original crab artwork before we tell you more about our original research. My name is AJ. I work in ocean education, both as an OSU employee and also as a PhD student.
Hi, my name's Omarius. I'm a volunteer at Siletz Valley High School. No, I'm a firefighter volunteer, and I go to Siletz Valley High School.
This entire project was motivated by getting high school students from Siletz to Hatfield, to the campus, to experience what it's like to go through this process of doing research for the first time. So we use this this framework of explore, experiment, and present to both inspire and support them as they went through it for the first time. What's unique about it is that these students don't do projects that are designated by an advisor. They design their own projects.
I was a part of a group of 9 students who luckily got the opportunity to do this. We first started exploring new places all around, just going everywhere. We first started with building ROVs, going left, right, up, down, picking up silverware. The biggest problem or the hardest part of building them was probably making sure that they floated, but they didn't hit the bottom of the tub that I was in. Glorious AJ bought us tickets to go whale watching. As soon as we got off the bay, it was extremely foggy and everything. I threw up.
You're not alone, Omar. We definitely, by the time we were done with our adventures, we needed some help. You might recognize some friendly faces on here. There's a couple of them in the audience tonight. These are graduate students who acted as mentors, and we're really able to enable a very wide variety of projects. So I'll just tell you a few of the stories from the students who designed things. This was a pair of friends who really mostly just wanted to catch crabs and be outside, and ended up designing an experiment about behavior. This was a team of brothers. They are not twins, but I called them by the wrong names for about a month, who really wanted to use local Oregon materials to clean up after oil spills. They tested a lot of stuff. And this boyfriend-girlfriend duo actually did pretty well. They really were fascinated by the tides, especially the king tides, which we had fantastic king tides over the winter. They designed a device to capture that energy and use it to provide power.
I couldn't start or finish my project without the help of my mentor, Abby, and an engineer, Drummonds. I first wanted to build a artificial coral reef that would monitor different aspects of water quality. When I came to Drummonds, I decided what type of plastic would be the best to use. He answered with, I don't know. I decided to go from there and choose 2 different types of plastics. I left them in water, salt water and fresh water for 7 weeks, and I tested how strong they were, how much force they could take. I almost broke my kneecaps during that experiment.
Couldn't figure out how to embed the video, but here we go.
I just wanted to take this opportunity to thank everybody who helped us during this project, anybody who supported it in the slightest. It was a great opportunity, and I might want to go into this career.
We all hope so. So I took Omar and the other students to compete at the Central Western Oregon Science Expo hosted at University of Oregon. And, even though they were there, they still had a great time, not as great as OSU, but they got to present their posters. I told Omar I was going to do this slide so I could brag about him. He did amazing. He won 2 prizes that came with some money, including the most prestigious prize at the fair. Yes. Yay! While they were there, the cycle begins again, and they also got to explore some amazing things that U of O has to offer, including the sports science facility and the Natural History Museum. So, to finish up, our final message for you is that
Just take the opportunity to.
We're overwhelmed by how many things we want to tell you. There's a lot of potential in the students that are here, and we hope that in the future, when I send out mass emails begging you to be a mentor, that you might consider joining us in our adventures. Thank you.
Wow. Wow. Just wow. Our next speaker is Sarah Hutton, who is a PhD student in Fisheries, Wildlife, and Conservation Sciences. Oh. Sarah Hutton is advised by Suzanne Brander and received the 2022 Pratt Award.
Hi, my name is Sarah Hutton, and I recently finished my PhD in Suzanne Brander's lab and was in the Department of Environmental and Molecular Toxicology. For this presentation, I'll be discussing my work on micro and nanoplastics under both unweathered and weathered conditions in larval inland silverside fish. Micro and nanoplastics come from a variety of different sources. They can come from both primary and secondary sources and encompass a size range from anything less than 5 millimeters for microplastics and less than 1,000 nanometers for nanoplastics. They've been found in almost every environmental matrix in which they've been looked at, and the leading hypotheses for their effects on organism are through food dilution, internal and external damage, and oxidative stress. Micro and nanoplastic studies often use these polystyrene spheres instead of those plastics that are more representative of the environment. As you can see from this figure, it's mostly fragments and fibers that are found in the environment as opposed to spheres. So for this study, we used microfiber, as seen here, and particle fragments instead of spheres. And this is an example from the tire wear particles that were also used in this study. We had 2 hypotheses. First being that weathered micro and nanoplastics will have greater toxicity than their unweathered counterparts, and that gene ontology and KEGG pathway analysis following RNA sequencing would provide insight into potential mechanisms of toxicity. So we had 3 plastic types: tire particles, PLA, and microfiber. Tire particles and PLA had both a nano and a micro size fraction, and all of them had a weathered and unweathered treatment. We exposed inland silverside larvae for 21 days, and then at the end of the exposure period, collected data on growth, behavior, and gene expression, which was then used for gene analysis for gene ontology and KEGG pathway analysis. So the first set of results are our growth index following the exposure. You can see that the unweathered polyester microfibers had the only significant effect on growth. This may be due to reduced feeding or false satiation of the organism exposed to those fibers. We looked at a behavioral response called thigmotaxis, which is a measure of anxiety. And as you can see, the weathered microfibers in this case had an increase in anxiety similar to the other nanoparticles in this experiment. However, we saw mixed results with PLA, and we did not see any differences between the weathered and unweathered treatments in the tire particles. This effect where the microfibers that were weathered had a similar effect as the nanoparticles was also seen in the gene ontology results, where we saw very similar expression of terms between these two treatments. And one of the reasons why the weathered microfibers may be similar to the nanoparticles is because we used UVC to weather them to represent wastewater treatment plants. And this process can cause small nanoparticles of the polyester to break off, which may be one of the reasons why we see similar effects in nanoparticles. So to kind of expand on some of these GO terms, we saw a lot that were related to muscle function, cell signaling, and osmoregulation. Effects on muscle function are important because it could lead to changes in behavior, but also because muscle disease is not normally apparent until later in life. And so we would not be able to see those sorts of effects unless the experiment was carried on until adulthood. In terms of cell signaling and osmoregulation, this is important because these inland silverside fish are euryhaline, which means that they can exist across a wide range of salinities, but effects on signal transduction and osmoregulation may impact their ability to move throughout the estuary and regulate their bodies as salinity changes. So to conclude, we found that weathered microfibers induce similar effects as other nanoplastics. And this is in agreement with other studies. We will need more longer term and multi-generational studies to understand how these effects could persist into adulthood, such as muscular disease. And in conclusion, we did not see that weathered or unweathered nanoplastics were more or less toxic than each other, but gene ontology and KEGG pathway analysis did provide insight into some new potential mechanisms of toxicity. So with that, I would like to thank you for your attention. I'd like to thank the Ivan Pratt Memorial Scholarship for their funding and all of the collaborators and help I received for this project. My email is below if you'd like to contact me with questions. Thank you.
And our next speaker is MK English, who's a master's student in microbiology, advised and mentored by Ryan Mueller, received the 2021 Robinson Award.
Okay. Hi, my name is MK. I'm actually a PhD candidate, but same thing. In Ryan Mueller's lab in microbiology, I was awarded the Anja Robinson Fellowship. And today I'll be telling you about how iron acquisition may play a role in probiotic efficacy. So oyster aquaculture is a really big industry, and it's projected to get even bigger. In the Pacific Northwest in particular, shellfish aquaculture brings in over $300 million every year, which is a lot. And it all starts in a hatchery where eggs are fertilized and grow into these larvae, which then raised to spat, sold to farms. Oregon is special in this regard because Oregon supplies a lot of the larvae for growers up and down the West Coast. However, these larvae in their early stages of their life cycle are susceptible to pathogen infection. So a pathogen can get into a clutch and wipe out the entire thing. And this is obviously economically really devastating. So to get around this, aquaculture farmers have started using probiotics, which are these live organisms that provide some benefit to the host. And this benefit may be disease resistance. So we wanted a probiotic for oysters. So we looked for a bacteria that could do two things. First of all, reduce mortality from the pathogen. And then second, provide a secondary benefit like improve oyster health or size. So we started looking, and we found a probiotic that on a plate assay, it didn't let the pathogen grow anywhere around it. So it circled, and then all the blank space around it is where the pathogen couldn't grow, which is awesome. But does it work in an actual live oyster? Yes, it improves oyster survival. We infected a bunch of larvae with this pathogen and then also gave them probiotic. And as soon as we added the probiotic, the survival rate shot up. So now we've got these two pieces of evidence that this probiotic has anti-pathogenic and then pro-oyster effects. But we've got absolutely no idea why. So we started to try to figure out how to solve that, how to answer that. So we had two hypotheses. First was that the probiotic was outcompeting the pathogen for either nutrients or space in the oyster gut. A second hypothesis would be that it's secreting some kind of molecule that is toxic. So to investigate this, we use transcriptomics, which is the study of gene expression. So when you look at the genes a cell is expressing, you get an exact replica or an exact model of that organism's activity at that moment in time. So we did this on some plates. We set up a pretty simple experiment where we plated the probiotic and then the pathogen, and then one plate was a co-culture. So it had both species competing for the same nutrients in same space. And what we found on that plate was that the probiotic cells radiated from the center overgrew the pathogenic cells and then created this frontier. So we collected from the frontier and then sequenced the genes to see what the probiotics were doing. And we found two pathways that were overexpressed in the co-culture consistently. So the first pathway is chemotaxis. The second one is iron sequestration, and I'll go into both of those. So first of all, iron, everything on Earth needs it, but it's really rare, especially for bacteria. So what bacteria do is they send out these molecules called siderophores that bind the iron, bring it into the cell in a form that it can use. Second, chemotaxis is a cell physically moving towards some nutrient source. So in our plate assay, that would be the probiotics depleting all the nutrients in the center and then searching outwards for things they haven't eaten yet. But how do these two fit together? My hypothesis is that the probiotic is able to move towards the iron and take it up better than the pathogen. And this leaves a pathogen without a vital micronutrient. So what else is involved? So we started to answer this. The probiotic outcompetes the pathogen for iron, perhaps. But it's important to remember it's a pretty complicated system. And there's a lot of other things going on. So in summary, probiotic may be outcompeting the pathogen for iron. A lot of other things going on. And we hope to answer those because I need to graduate. So big thank you to everyone on this screen. This would not be possible without help from a lot of people, including the Andrew Robinson Fellowship. And thank you all for listening.
Great talk, great talk. All right, and our next speaker is Spencer Lunda, who is a PhD student in the Comparative Health Sciences program, mentored and advised by Carla Schubiger, who received the 2022 Anya Robinson Award as well. And it's a recording.
Hi, everyone. My name is Spencer Lunda. I'm a PhD student at Oregon State University, based here at Hatfield Marine Science Center. And today I'll be talking to you about some research I've done on deprecation of Pacific oysters with the goal of reducing Vibrio parahemolyticus. So the Pacific oyster industry. The west coast of the U.S. contributes 1/3 of the total U.S. shellfish production. This is mostly oysters, which was valued at about $105 million in 2018. But a potential issue here is that oysters can harbor the bacterial pathogen Vibrio parahemolyticus. So what is Vibrio parahemolyticus? Well, it's found globally in marine environments. It's a pathogenic bacteria that thrives above 15 degrees Celsius, and it's a major cause of foodborne illness, in particular from undercooked shellfish. It is destroyed by cooking, but some people like to eat raw oysters, like myself. So how can we prevent Vibrio parahemolyticus infection in humans that are going to be eating these oysters? Well, there's a few different post-harvest processing methods, including freezing, high hydrostatic pressure, and deprecation, which I'll be talking about today. So why use deprecation as opposed to other methods? So other processing methods can alter the flavor and texture of oysters, and for people that like to eat raw oysters, that's not ideal, in particular since these methods kill oysters, while deprecation does not. So we can have an oyster that looks like that one in my hand there. And now, what is the process of deprecation? This is basically just holding shellfish in clean, sterile seawater and allowing them to naturally purge over time. It's been used for decades to reduce pollution-associated bacteria like E. coli, but is not used as much for Vibrio. So this here is showing a pilot scale deprecation system like we have at Hatfield. So this red arrow at the top is just indicating the direction of seawater flow. On the left here, we have filters, a temperature regulator, which controls the temperature of the seawater, and a chiller unit. So the seawater flows through that blue pipe from the chiller unit, and this UV sterilizer here flows up and then falls down into these holding chambers where the oysters are sitting. And then water flows from there down into a holding tank and then recirculates through that entire system. So to set up a deprivation experiment, first we have to inoculate our oysters with this bacterial pathogen of interest. So you'll place the oyster in seawater, add the pathogen to that water, and then just allow the oysters to sit overnight and take up that pathogen. So for deprivation to occur, first we fill the system with seawater and set it to its desired temperature and just allow it to chill for 24 hours. Then we add our oysters randomly into holding chambers and allow them to just undergo deprivation for the next 5 to 7 days. So then to quantify Vibrio paralyticus in these oysters, we'll take an oyster out of the deprivation system, we shuck it, we homogenize that, and we plate it on selective media that will only allow Vibrio to grow. After 24 hours, we come through and we count how many colonies are present, and that'll tell us what the Vibrio concentration is. So these are just some initial results of deprivation research that I've conducted. So basically, this is 3 different species of oysters that we allowed to undergo deprivation, and what we saw is that we were able to get these reductions in Vibrio paralyticus, but not quite what we were hoping to achieve. And so we observed a few issues with reducing VP in oysters. One is the clearance rates can be inconsistent between trials. Another is that lingering bacteria just don't reduce after those first 1 to 2 days. And also inoculation of the oysters can be variable. So to kind of further understand and get a better idea of what's going on here, something that could be of interest is, do feeding oysters during inoculation increase the uptake of Vibrio paralyticus? And then also, does the microbiome of oysters change over the course of deprivation? So do we see different bacteria present in them? So some hypotheses. Feeding oysters during inoculation should increase the Vibrio paralyticus uptake by increasing the oyster activity since there's more food present. And also, as deprivation occurs, certain bacteria should be reduced in abundance while others should linger. To answer that first question, I conducted an algae inoculation study where I fed oysters algae during inoculation and determined if that increased their uptake of VP. I also monitored the algae's density in the water after inoculation to see if it had been reduced. And so this is just showing how that worked. So I cultured algae in flasks, added that to containers of oysters, to which I also added Vibrio paralyticus, and compared between tanks that received algae and tanks that received no algae to see, did I see a difference in bacterial concentrations. And so when I looked at the concentrations of Vibrio in those 2 different treatments, what I saw is that there was no difference in bacterial density between the 2 treatments. So that was in both oyster tissue and in the liquor, which is the liquid inside of the oyster shell. So going forward, I'd like to look at the effect of deprivation on the microbiome, or the bacteria that are present within the oysters. So what I expect to see is that over the course of deprivation, overall bacterial abundance will decline, but also certain taxa will remain while others disappear. So thank you for listening, and I'd just like to acknowledge that this research has been funded by a 2022 Anya Robinson Fellowship, as well as a 2022 Mamie Markham Research Award. So thank you for your time.
All right, and our next speaker is Hillary Thalman, who's a PhD student in the Fisheries Wildlife Conservation Sciences Department, advised and mentored by Jessica Miller, received the 2022 WIC Award, as well as the 2022 Marianne Bozo Award.
Yeah. Hi, everybody. My name is Hillary Thalman, and today I'm going to be talking to you about whether or not carbon-stable isotopes in Pacific cod otoliths can be used as a metabolic proxy. So many of you may know about the marine heat waves that we've had over the last couple of years. Those had giant impacts to the ecosystem, widespread declines of a lot of species, but the one that we're particularly interested in is the decline of Pacific cod. And we know that as we get these really warm temperatures that you see in a heat wave, you would expect that fish metabolic rate would also increase. It follows a pretty predictable relationship with temperature. And in these situations, a fish either needs to consume more food or move to cooler waters to survive. And so metabolic rate as a whole is just a measure of the energy expenditure of the fish. And the metabolic rate that you measure in the field is a combination of the standard metabolic rate, which is that baseline rate of the fish, plus digestion, plus any activity that the fish is doing. But the problem with this is that measuring metabolic rate in the field is really difficult. You can do it pretty well in the lab and you can estimate it with models, but getting that field measurement is a challenge. And one way that we might be able to do this is with stable isotopes. So stable isotopes occur when you have an element that has more than one stable form. So for carbon, we have carbon-12, which is the most common form, and carbon-13 when we add an extra neutron. And we can trace biological processes with isotopes. And the isotopes that we're interested in here are found in fish otoliths. So fish otoliths are calcium carbonate structures in the inner ear of fishes, and they're interesting to us because they can provide information about the environment of the fish. The elements in the environment can be incorporated into that otolith structure. And we're particularly interested in carbon. There's only two sources of carbon that the fish can have in its life, carbon from its diet and carbon from the water column. And these have very different carbon-13 values. The carbon value in the diet is a lot lower than the inorganic carbon in the water. And these both get incorporated into the otolith matrix. So you have a combination of dietary carbon and inorganic carbon. And so if you measure D13 in carbon-13 in the otolith, you can hypothetically get a lower delta 13 value for a higher metabolic rate. There's an inverse relationship there. So what we're particularly interested in is to see if this works for Pacific cod. It's been shown to work for other fishes, but not for the species or this life stage. So can otolith carbon-13 serve as a metabolic proxy for this species? And to do this, we did two different lab experiments. The first experiment was a more moderate temperature experiment that you'll see in a minute, where we raised fish in 4 temperatures that are pretty typical of what they'd experience in a normal year. And the second experiment, we added in these really extreme temperatures that the fish is likely to experience in a marine heat wave, which you'll see in a minute. And each of these fish we reared for about a year in these experimental temperatures. We tested the water and the food for carbon-13 each week. And then after their rearing period was up, we put them in swim tunnels. And swim tunnels are a little bit like water treadmills. The fish is swimming against a water current, and you can measure their oxygen consumption in that swim tunnel to get at their metabolic rate. And then you can take that rate and compare it to the otolith carbon. So we take the otoliths and we micromill them with a tiny drill press, and we get tiny amounts of carbon powder from the otolith, which we then run for stable isotope analysis. We put that all into a stable isotope mixing model to get at the metabolic carbon in the otolith. So what did we find? So this is the metabolic rate from the swim tunnels for our first set of moderate temperatures. And what's really surprising is there's not very much variability with metabolic rate in all these different temperature treatments. And that really surprised us because it ought to increase. And when we add these really extreme temperatures, we again don't see variability in metabolic rate within the temperature treatments, but we do see a big stepwise increase between those two experiments. And if we look at the carbon as it relates to these fish, the first experiment, we don't really see a variation in carbon in the temperature treatments either, which if this is a proxy for metabolic rate, that is matching what we see in the metabolic rate, but not particularly promising as a strong indicator of metabolic rate. The second experiment, we are processing those otoliths right now. We don't know what we'll see, but we potentially could see the same stepwise increase that we see with the metabolic rate. So these results were really surprising. We didn't expect to see no relationship with temperature and metabolic rate, and they're so far inconclusive for the use of this as a proxy for this species. But this is still active research, and we're working on getting some new results soon, and I look forward to sharing those with you soon. So thank you.
Okay, great talk. And our final presentation today is from Keiko Nomura, who is a PhD student in the Marine Resource Management program mentored by James Watson, who received the 2022 WIC Award.
Hi, everybody. My name is Keiko. I'm a PhD candidate in geography, though I was lucky enough to do the Marine Resource Management program a few years ago. And I'm going to talk to you today about international fisheries conflicts and how we might be able to study high-risk regions and actors where these could emerge. So I want to start by imagining the ocean. Let's say this is the Pacific Ocean. This place is huge, And yet the fishing activities and the interactions that take place out here between fishing vessels can lead to conflicts, and those can actually scale up to have international relations implications. And this is actually the reality of fisheries conflicts today. Globally, these events are increasing, notably between international actors, and this has wide-reaching implications for fishers' well-being, economic vitality, fishery sustainability, and increasingly geopolitical relationships. These are really diverse events. They can range from verbal disagreements to policy changes, all the way up to physical violence and military involvement. Yet the drivers and the conditions underlying these are largely difficult to study, and so mediation is hard. However, one driver is spatial competition. And indeed, as global fishing activities expand over the world's oceans through distant water fishing fleets, the geography of where there is spatial overlap and where related conflicts can emerge is fundamentally changing. So my project takes a look at fishing vessel tracking data to look at spatial competition across the Pacific Ocean. And we can use this vessel tracking data combined with ecological measures of diversity to understand where there is diverse international fishing pressure in different parts of the ocean. 55 countries fish here, and we find the tropical Pacific is by far the busiest, particularly these areas beyond national jurisdiction, high seas pockets adjacent to national waters, but also international waters too. So this competition is spanning many jurisdictions. We can even take a network analysis approach where each of these circles here represents a different country, and they're connected if they share ocean space. The thicker the line, the more ocean space. This allows us to visualize who might be spatially dominant and specific pairwise relationships. And we see that there are easily a few major dominant players in this area, namely Taiwan, China, South Korea, Vanuatu, and Japan. So this gives us an idea of spatial crowding, which is great, but we want to know a little bit more. We want to know how these countries have historically gotten along. Say you and I share a room and we're friends, that's cool. But if you and I are not such good friends and we share a room, maybe that's a risk for conflict to emerge, right? I'm sure everybody has had a bad roommate before. And international relations research actually calls this enduring rivalries. So we can look into historical fisheries conflicts to understand who those enduring rivals might be when it comes to fisheries related conflicts. And we can then prune down that initial map that will show up now to then take a look at only areas of overlap where countries are sharing if they also don't get along. So we're kind of looking at where these bad roommates are hanging out on the ocean because we think maybe that would mark an elevated risk of conflict. Now it's very similar to that other map, but smaller in area, as we would imagine. And the Pacific Ocean emerges again, and particularly those boundary conditions right along the national waters and between the high seas come out. In one instance, in a high pocket area, we had 13 risky pairs of countries sharing a really small ocean space. Similar to before, we can also take a look at the network approach too, to look at very specific international relationships. Again, the thicker the line, the higher the risk, and that's based on the average conflict intensity and the total amount of area shared. We see similar players emerge here: South Korea, Japan, China, Taiwan, Vanuatu, and also the United States because of the amount of fisheries conflicts that we've gotten into with other countries. So the main takeaways here are that global fishing is now probably more than ever a global enterprise occurring across jurisdictions. Conflicts can arise between countries that are otherwise geographically distant from each other, and this can be surprising. And this is really relevant information for stakeholders. Regional fishery management organizations are prominent in this area. Maritime security agencies would like to know this as well. So to recap, we've taken the lenses of spatial competition and enduring rivalries to take a very systems level perspective, international in scale, to think about conflict. And this is really useful when you're thinking about a large, globalized, complex socio-ecological system, thinking about systemic risks, but it certainly trades off for context and detail. So future work can look at more specific conflicts over species, intensity levels, and within specific actor relationships. Regardless, though, a variety of approaches is going to be needed if we're going to proactively address the systemic risks that are arising from international and evolving fisheries. This is especially true given the wide-reaching implications it can have for the social, environmental, economic, and geopolitical contexts. So with that, I'll say thank you, and thank you to the Markham Symposium and the Bill Wick Marine Fisheries Award.
right, so great presentations. Again, another round of applause for all the speakers. So just want to highlight that graduate and undergraduate education is at the heart of HMSC's mission to improve scientific understanding of marine systems, coastal processes, and resources, and applying this knowledge to social, economic, and environmental issues. These student awards and scholarships directly support our students as they pursue their research goals. They also recognize and validate students' outstanding research efforts. Thank you for joining us today. Please join me again in congratulating these awardees. I also want to make some special announcements, acknowledgements, and recognitions. Shannon Reed, Cinnamon Moffett, Katie Erdman, Miguel Medrano, Helen Bretan, and Tammy O'Connor for making this year's symposium a tremendous success. I also wanted to let last year's award winners know that if you'd like to collect your posters, they're available in the atrium. Thanks for letting us display them for this last year. Thank you all for coming. Now we'd like to invite the awardees, their advisors and mentors, our special guests, and summer interns to dinner on the second floor of the mezzanine. And a reminder to please, please, please submit your ballots. Thank you for joining us today.
For everyone online, thanks for being here.